In this video, I'm going to show you how you can perform tracking with TrackMate. So we're going to use this very simple image sequence of a C. elegans embryo, where it comes from a 3D volume, 3D stack over time, but here is just the maximum intensity projection. It's going to be fine for what we want to do. So as you can see here, we have, let's start with these two cells that divide, and then each of them divide into two daughter cells. And then it's going to keep going here. We see it starting the next division, two of them. As you can see, we also have some biological objects that are, so here at the pole of the cell. I'm not sure exactly what it is. It might be here, we see it might be mid-body, or I don't know exactly. Clearly not cells. So they're going to disturb the tracking. So we see how we can deal with that. So we're going to use TrackMate, which is a very easy to use plugin within Fiji, but allows to do a lot of interactive processing for tracking. So I'm going to open it. If you go to plugins and then tracking, you're going to be able to find TrackMate here. All right, so it asks you a few questions about the pixel dimensions, voxel depth. So everything was in the metadata, so it should be fine. So here it might be important to look at this to make sure that you have time and z dimension that are the right ones. So sometimes this is going to be saved in x, y, and z instead of x, y, and t. But in that case, TrackMate is going to ask you if you want to swap them. But if you have both z and t, just make sure that z and t are actually well defined here. Then you can go to next. So that always works like this with TrackMate. You go through a window, you do everything you want to change in there, and then you go to next. So the first step is to detect your object. So here we want to detect the cells. And as it's fairly easy here, we're just going to do a thresholding. So with the new generation of TrackMate, there are many things you can do, including using deep learning approaches like StarList or cell polls, which are very, very powerful. But here, we're going to use just the thresholding. So you could also use log of the dog, which are really good when you have objects of the same size. Here it wouldn't work because when the cells divide, they are smaller after division, and then they increase in size. So that wouldn't work. So I'm going to just use the thresholding detector. And then, so you can define your threshold and preview. So if I preview here, I see that I have those 4 objects. It might be a good idea to go to other stacks and to other frames, and actually to frames where you have low intensity. So you're going to see which should be the the threshold. Here, obviously, I don't detect them, so I need to decrease this. So let's say to 800. Seems to work. And so let's keep going. Now we have detected the cells. So you do next and then next. And again, here we need to go to the next step, which is going to be filtering the spots. So I have a bunch of filters. I'm just going to remove all of them. Filtering spots is very powerful. It's just that if you have too many objects, you can remove a few of them, but might, later on mess up your tracking. The better the segmentation is, the easier it's going to be to do the tracking. It's really important to have very good segmentation to start with. And so filtering the spots that do not correspond to what you want to track is really, really interesting. So now that the the detection part has been done, I have my spot all along. And so maybe here, sometimes we have things that do not, if you look at this, here obviously this is larger because they are both together. And this is going to be probably the smallest size for my cells. But then if I keep going, I have some objects that might be smaller than this that I don't want to have, like this one. So maybe I can put a filter on the size. So let's just go to area. And so you see this has been filtered out. So here, so you might think, okay, I don't have this thing, so that's good, but I don't have these ones as well. Then I have them all the time. I don't have this one. I don't have these two. So maybe, maybe it's better to keep having this one. So I'm going to just try to find a threshold where I have them. So I'm going to, here I have those two, but I don't have those two, which is exactly what I want to do. I know that these 4 objects correspond to cells. These 2 don't. And then I can just look at my sequence, see if I have the cells all the way from the beginning to the end. So I need to go from the beginning. I have them. And so you see here, I still have this thing which doesn't correspond to cells. I have this thing, but I don't have this one, which is pretty neat. I have this one. I don't have this one. So you see, it's going to be then probably easy to get rid of them. And I have the cells all along, which is a good thing. So I'm going to go with this. I could filter spots based on other features. So obviously you could use the intensity, but here, as the cells are dividing, they go from very bright to not that bright. So that might not be very informative, and I won't be able to discriminate between this and this based on intensity. So I think I'm just going to go there, and I'm going to track my cells, and I'm going to use simple lab tracker first to show you how we can interact and manually change the tracks at the end. So when you use the simple lab tracker, you have 3 parameters to define: the maximum distance between 2 frames, so let's say 15. I think 10 would be fine, but let's just say 15. You can have some gap closing, meaning if you have an object that disappears, then you're going to try to look at it in the next frames. And in that case here, it would be it can disappear for 3 frames, and I'm going to look in the neighborhood of 15 micrometers. The way we did the detection here, we never miss the cells, so we don't need to do gap closing. So I'm going to just put this to 0, so it's not going to do any gap closing. Just a linking between 2 consecutive frames. And now, as you see, so we have those tracks. You see the different colors. I have 4 tracks for my cells, corresponding to the 4 cells at the end. All right. And I have this small track. So first thing I want to do is to remove this track. It's already done. I'm going to remove this. I want to remove this track. So I can filter out tracks the same way I filter out spots. And here, this track is actually pretty small. So if I just, because after this point I don't detect any spots, so there's no track in there. It's just at the beginning. So if I just filter out, depending on the number of spots in the tracks, if I don't put, if I go to 0, I have this, but if I just go up to 4 spots, now I don't have any track. And we can look at the histogram. Basically, we have 2 tracks that go from the beginning to the end. We have 2 tracks here that start when the cells divide, and this track. So I can, that would be easy to just set the threshold between this track, which is meaningless, and the other tracks. All right. So we can look at the different things, the spots and the tracks with different colors. Now that my tracks are well defined, maybe it's a good idea, so it depends on what you want to do, but you can also give, so you can give the color, the same color for the spots that belong to the tracks. So if I go to color spots, I define it as track, it's going to be track index. You see here it's already track index. Now my spots have the same color than the tracks. See, so this is purple spot and track, blue spot and track. When the other appear, they have the same color. Something also, if you don't want to have the entire track from beginning to the end, especially if you have many objects, you can, for example, just show tracks backward in time. Means that the tracks start tracing when it appears. So I don't have any track. I have beginning of the track, and you see, just keep going, and then you have the other ones, and you can fade the range. So if I fade the range at 5, I'm just gonna have the track for 5 previous frames. So you can play with that. Right? Now, as I did a simple lap tracker, I couldn't divide the cells. So here you see when the cells divide, we have new tracks, which is not what we want. So I can change that manually. If you go to track scheme, then you have a lineage tree, and I have just 4 tracks here. And obviously, when we look at the colors, we see that the blue divides after the division. We have the blue and the orange. So it's interactive. If I click on the first frame for the orange track, it shows me which spot corresponds to this one. I have all the features that are associated to this spot as well. And the good thing is that I know that if I go at the same time for the blue track correspond to the other one, if I go the frame before, it's actually the mother cell. So here, what I want to do is to connect this spot to this one to show that this track actually, so it starts from the beginning and then it divides into 2 cells. And it's really easy to do it with TrackMate. You just click on this and then you can just link the cells you want to link. You can lay out, and now those 2 are linked together. And you see now they have the same color because it's the same. That's the same track for both of them. It's just that it divides at that time. And I can do the same thing here if I so I'm going to unclick there so I can click there. So this is the first frame where I have 2. This is the frame before, so I need to connect this to this. Alright, if I do layout, now I start with 2 cells. When they divide, it's still the same tracks, but I just have 2 cells. That's exactly what you want to have in your lineage tree. And so here we manually did it. Alright, you can go back to TrackMate. What I did there, I can do it directly during the tracking if I allow track splitting. So I'm going to go back in time. And instead of simple lap tracker, I'm going to select the lap tracker. All right. With a lap tracker, what you can do, you still have a frame-to-frame linking. I'm going to keep it as is. You have the gap closing. As I said, we have all the spots, so I don't need gap closing. You have the track splitting, so I'm going to allow this, so the track to split. You also have a track merging if you want. So if you know that you have objects that can merge, this can be very useful. The other thing you can do is to add some penalties. So if you have objects that have the same intensity over time, for example, you could add a penalty and then use the average intensity, mean intensity in the channel, for example. So here, the intensity is changing because of the cell divisions. It's not going to help, and tracking is not hard, so I don't need that. But that can be useful. So I have same frame linking than before. I allow now track splitting. So let's see if it's able to do it by itself. All right, and as you see now, I still have this track corresponding to this one, which is filtered out here. So if I go down to 0, I would have it, but I don't want it, so I'm just going to go there. And if I go back in time, just to show you, it wasn't shown because I wasn't there. But you see, if I have this track, but I want to filter it out, just going to define the threshold. And now I have two tracks that are same length as a sequence, and they start from there. And when they divide, the tracker was able to realize that it was the same cell that was dividing. So here, I don't have to do anything. If I go to the next step, I can open the track scheme, and I only have two tracks. And just a couple of additional things you can do. You can plot some features. So let's do it for the spots. If we plot mean intensity and let's say the size, area, you can click on plot features. Interestingly, you see that the area is increasing, and then so to maximum and then decreasing corresponding to cell division. And then you see you have two tracks in blue and in red that all again increase up to some decreasing. At the end, we see that it's the new division that is starting. So the area goes from large to small again. If you look at the intensity, you have some correlation. You start with very high intensity for both of them, and then when the increase in size, intensity is decreasing and is increasing again slowly up to the division, where it's really bright after division and decrease again. So you see that the intensity increase in sequence with this. So you have first the size and then the intensity. So that looks like what we see in the sequence. Obviously, we could do other things. So you can look at link features. So let's look at the speed, for example, the displacement. And again, you have large displacement, maybe larger displacement when you have the division, because after division, they go further away. It's not that drastic. We could maybe look at other features that might be more meaningful. You can also plot features related to tracks. So if we look at the total distance traveled, for example, in that case, it's only 1 point per track, which makes sense. And it looks like, you know, the first one, which is, I think, the purple one, this cell goes through a larger distance than the other ones. Of course, here with two tracks, not very informative. If you have many objects, looking at individual features for each track might be also very useful. So that's, you know, one way to look at your different features. You can then do a few other things. If you want to save your overlay, you can execute it. You here select what you want to have. If you just want to have the tracks, you can do it. If you want to have the tracks overlaid on the image, which is the first and last frame, and just create this with the overlays. See, you can save it to show your results. There's a few things you can do it. It's pretty self-explanatory. I'm not going to go through that. You can save it as an XML file, so then you can open it again with TrackMate. Actually, it's if you go to tracking, you have load TrackMate file. So if you save your TrackMate file, you can then load it, and it's going to load everything for you. You can also save all the information about the spots, about the tracks as well. Let me check. If you go to tracks here, it's interesting because you have everything that is embedded here. You actually have 3 tabs. You have one for spots, so each line corresponds to a spot, and I think if you click on this, you can, it's interactive. You can see which one is there. So you have a spot ID, then corresponding to which track ID, and then all the features that are associated with the spot. You have edge information. So edge information tells you from which spot to which spot. So again, this one is not very explicit. So here it's between those 2 frames, you see. And then you have features associated with the different edges. And then you have your tracks. So here we only have 2 tracks. You have the IDs and number of spots in each of them. So we have one more spot for the red track, because at the end, no, I don't even know when we have. No, it's at the division. Sorry, it divides. Here we have 2, and we still have 1. So that's why we have one more spot. So, and you have all the features associated with the tracks. And so then when you combine all this information, you can know which spots are in the track, when they do divide, and so on and so forth. And you can export it to CSV as well. So you have all the information that might be useful for your analysis.